everybody, let's move on to part 2 of chapter 4, still dealing with genetics and how the cell functions. Now, we're going to dive into DNA replication and the cell cycle. And when we look at DNA replication, you know it's very, very necessary prior to cell division and is accomplished through complementary base pairing, which we went over already. Now, we already went over the law of complementary base pairing, where we predict that the base sequence of one DNA strand if we know the sequence of the other. Okay, and it enables a cell to produce one strand based on the information in another. Now, when it comes to DNA replication, we're going to go through a number of steps. This diagram is in your textbook on page 127, so you guys can follow along. But the information for this particular diagram is after this particular slide. So I'm just going to go through this diagram and then I'm going to skip through to the next process, the next procedure. So I'm not going to go through each slide because the information is already there. So we're here. This is somewhat DNA replication, the whole 9 yards about this particular diagram. So I'd rather have you look at the diagram and talk about it than us going through the other slides. Alright, so the first step, when we talk about DNA replication, the first step is the unwinding of the double helix from the histone. So hopefully you remember, you remember the different parts of the DNA, what is made up of, the histones, the chromatin, the centromeres, so everything will make sense. Now, so that first step, as I said, is the unwinding of the double helix from the histones. So here's the parental DNA, here's it unwinding. Then we have an enzyme called the DNA helicase. And this opens up a short segment of the helix. And it's going to expose the bases as you see them exposed right now. Okay, all of those bases are exposed. Now the point where the DNA is open is called the replication fork. Okay. Now molecules of the DNA polymerase match the exposed bases with complementary free nucleotides. Now the two strands are copied by separate DNA polymerase. So here's one here, here's one over here. Those two polymerase molecules move in opposite directions. So you see the top DNA polymerase is moving towards the left. The bottom ones are moving towards the right. Now one polymerase makes a long continuous strand, and the others they're going to make short strands moving away from the fork. So these are going to make the short strands. This one's going to make the very long strand. Now, while these two are moving away from this replication fork, they tend to join together by the DNA ligase. So yes, they're making short ones, those short DNA, but they come together with this DNA ligase at the end. Okay. Now the two new daughter DNA molecules. Okay. Those are synthesized from original parent molecule, and each daughter DNA consists of one old or conserved helix and one new newly synthesized helix. So here we have the old from the parent running through, and then we see the nice colored one here, which is they represent it as that new strand, as that new strand. So the daughter cell will have both the old and the new DNA strand. That's for both of them. Here we have the parent running through in the blue. Then we have the very short strands being replicated, and at the end we have our parent and our new strand, our old and new strand, forming our daughter DNA. Now, this process here that I'm referring to with the two new daughter DNA molecules, that process is therefore called a semi-conservative replication. And while DNA is synthesized in the nucleus, new histones are being synthesized in the cytoplasm and then are transported into the nucleus for combination with each new DNA helix to make new nucleosomes. Of course, you're going to have 1,000 polymerase molecules that are working together simultaneously on each of these DNA molecules. Now, let me move forward to the mutations. We talk about mutations. Of course, these are changes that we have within the DNA structure, and they result from replication errors or environmental factors. They say like UV rays or ultraviolet rays. Oh, no, same thing. UV rays or X-ray machines that may cause mutations within this particular DNA, which eventually causes diseases, illnesses. It can kill cells, cause cancer, genetic defects in, of course, future generations. So these things are called mutations. Now, the DNA polymerase may make errors in copying, but there are many codes of error correction. Now, some mutations have no effect because they do not change the amino acid for which they code. For example, TGG, which we saw in our chart. You know, it's in our chart back on on table, I think it's table 4.1, where you have the different amino acids. For example, our ACC and ACG, we still get the same amino acid. Or for GGA or GGU, GGG, it's all glycerine. So yes, the code may have the base code or the codon may have been changed, but it's still giving you that same genetic code. It's still going to give you that same amino acid. It's where it comes into what causes mutation is where you're having GGG, which is your glycerine. Then you have this mutation occurring, giving you GCA, which is GGA, which is your alanine or alanine. That's where the mutation comes in. Okay. So, now since 98% of DNA does not code for proteins, the majority of the mutations do not affect the protein structure. do not change the structure of the protein at all. So let's look at the cell cycle. You guys should remember this from high school. Remember mitosis, meiosis. All right. So this is what we're going to dive into. We're dealing with the cell cycle, and it's the life cycle period from one cell division to the next. And it has 4 main phases. It has our G1, our S. G1 is first gap phase. S is synthesis phase. G2, which is second gap, and then we have our M, which is a mitotic phase. So let's dive into each phase. Yes, you have an interphase which includes these subphases, but there are 4 main phases, which is 1, 2, 3, 4. 1 through 3 is basically dealing with your G1, your S, and your 3. And then we have this ESM, which is the mitotic phase. So we do have 4 main ones, but we have our subphases. Okay. Alright, so let's move on to what's happening with each phase. In G1, this is the first gap phase and is the interval between cell division and DNA replication. During this time, the cell synthesizes proteins, grows, and carries out its tasks in support of the body. Cells in G1 also accumulate the materials needed to replicate DNA in the next phase. S, which is the synthesis phase, is the period during which the cell makes duplicate copies of its centrioles and its entire nuclear DNA. In G2, which is our second gap phase, is a relatively brief interval during which the cell finishes replicating its centrioles and synthesizes enzymes that control cell division. It also checks replication for errors. This M phase, the mitotic phase, is the period during which a cell replicates its nucleus and then pinches into two to form two new daughter cells. Now phases G1, S, and G2 are collectively called our interphase. This is the time between the M phases. Now the length of the cell cycle varies depending on the cell type. Skin cells are going to divide rapidly, bone cells a bit slower, and skeletal muscle and nerve cells not at all. Now some cells cease to divide for days, years, or the rest of one's life. These are said to be in the GO or G0 phase, which is an inability to stop cycling and enter G0 and characteristic of cancer cells. Alright, so let's dive into mitosis. Cell division. Cells divide by two mechanisms: mitosis and meiosis. Meiosis is restricted to the production of eggs and sperm, and mitosis serves all the other functions of cell division. Okay, so mitosis is responsible for development of an individual from one celled fertilized egg. Mitosis brings about growth of all tissues and organs after birth. Cells that die are replaced by mitosis. Helps in the repairing of damaged tissues. And we have four phases of mitosis: prophase, metaphase, anaphase, telophase. These are our four phases. Alright, so of course I'm going to have my pictures, and the description is after, so you guys can, if you printed out the diagram or printed out pictures, the PowerPoint, you can follow along and make your own notes, that's fine, but I'm going to show you the diagram. When we look at prophase is here we have our cell, and prophase is a period during which the chromosomes are shortened. Our chromosomes, here are our chromosomes. The chromosomes are shortened and they're thickened. And they become compact rods, and then they start to align themselves in the middle of the cell. Now there are two chromatids per chromosome. Okay? Now the nuclear envelope disintegrates. Let's see if I can get this going. Here is my nuclear envelope. It starts to disintegrate. So you notice here, you don't see it here in the second phase. So it starts to disintegrate, and the chromosomes are released into the cytosol. So here's the cytosol. Now the centrioles, which are these here, find here, they sprout elongated, what are these called again? Oh, microtubules, that's right, called spindle fibers. So here are our centrioles, and here are our spindle fibers. And these are going to push the centrioles apart. Now some spindle fibers grow toward the chromosomes and attach to the kinetochore, the kinetochore, on each side of the centromere. Now the chromosomes are tugged back and forth until they line up the cell's midline. So now we're in phase 2. No, sorry, phase 2. The metaphase. With metaphase, this is in this particular period during which the aligned chromosomes, the chromosomes are aligned within the midline, and then here they are anchored by these particular spindles, the meiotic spindles, and then wait for the signal to split. In anaphase, this third phase is the cleaving of the centromere of each chromatid pair. So here's the cleaving, meaning start to in which you see it beginning to split, and you have, it's called cleaving. Okay. Now, after splitting apart, each chromatid is a daughter chromosome. One daughter chromosome migrates to one pole. Nope, I'm sorry. I'm mixing it up here. Alright, so when it says the anaphase, the anaphase is cleaving of the centromere. So here we have the separating of the centromeres of each of the chromatid pairs. So you have one pair going to your left and one pair going to your right. I'm very sorry. Now, after splitting apart, each chromatid is a daughter chromosome. So you have a daughter chromosome here, and you have a daughter chromosome on this side as well. Now, one daughter chromosome migrates to one pole, and the other migrates to the opposite pole. Now, migration is achieved by motor proteins in the kinetochore, and is crawling along these particular the spindle fibers. Now, the sister chromatids or daughter chromosomes are genetically identical to each other, and so the daughter cells that result from genetically identical Telophase here is the last phase of mitosis, during which the daughter chromosomes are clustered on each side of the cell. A new nuclear envelope forms, and the chromosomes begin to uncoil, while the meiotic spindles start to vanish. So you don't see the spindles anymore, but you still have the centromeres. Now, the cytokinesis. Cytokinesis, it overlaps this with the telophase, and it is the division of the cytoplasm into two cells. So here's your anaphase, where everything's beginning to migrate to other poles, one to the left, one to the right. Telophase, you have the cleavage forming of the cell, getting ready to separate. You have the dissolving or disappearing of the spindle fibers. But then, of course, to help complete or the overlapping of this phase is our cytokinesis, where they actually divide into two separate cells. That motor protein, myosin, it pulls on those microfilaments of the actin in the cytoskeleton, and it creates this cleavage. Now, the parent cells eventually pinch into two as the foyer is deepened, and the daughter cells enter into interphase. This is mitosis. Now, the timing of cell division and its inhibition are areas of intense research, which you should know already. Now, cells divide when a number of conditions are met. Now, when they grow large enough to have sufficient cytoplasm for two daughter cells, when they have replicated their DNA, when they have received adequate supply of nutrients, when they are stimulated by growth factors, when neighboring cells die, creating space for new cells. Now, cells cease dividing under other conditions. So they're going to start dividing under these three conditions. When they are snug in contact with neighboring cells, when growth factors are removed. And lastly, they come into contact inhibition is the sensation of cell division in response to contact with other cells. Now, when we talk about heredity is the transmission of genetic characteristics from the parent to the offspring. Now, karyotype is a chart of chromosomes arranged in pairs and by size and other physical characteristics, which you see in your textbook, I believe, on 131, 132, I believe is around there. Yes, 131, 132. You see it in your textbook, figure 4.17. Now, the two members of each chromosome pair are called homologous chromosomes. One is inherited from the mother and one from the father. Now, homologous chromosomes generally look alike with the exception of sex chromosomes, your X and your Y. The non-sex chromosomes are called autosomes. Now, a female has two homologous X chromosomes, and a male has one X chromosome and a smaller Y chromosome. Here's your chart that you see how they are arranged by size. And then, of course, physical characteristics. So you won't have, well, in a normal chromosome or karyotype chart, you won't have this one here, 17 paired with 1, unless there's some type of mutation or unless there's a situation going on, then you won't have 17 paired with 1 unless there's some type of mutation or a disorder going on that the individual may have. Alright, so any cell with 23 pairs of chromosome is said to be a diploid or diploid. I say diploid, diploid. Sperm and egg cells, however, are haploid, having half as many chromosomes. Now, sperm and egg cells, egg cells, and cells that become sperm cells and egg cells are called germ cells. Now, all other cells of the body are called somatic cells. Now, at fertilization, a sperm cell and egg cell, they fuse and they start to restore the diploid number. Now, each chromosome carries many genes. The location of a gene is called a locus. Homologous chromosomes may carry different forms of single genes called alleles at the same locus, at the same location. Alright, so this allele it produces alternate forms of a trait. Now, frequently one allele is dominant and the other allele is recessive. Now, a dominant allele, it masks the effect of a recessive allele. Recessive alleles are expressed only when present on both homologous chromosomes. Typically, dominant alleles code for a normal functional protein, and recessive alleles are for unfunctional variant. An example of a dominant trait is the trait of a cleft chin. There we go. Of a cleft chin, which is on, I guess, figure 4.19. Now you'll see that this, if it's a capital letter, it means dominant. If it's a lowercase, it's recessive. So this individual, which we already know who it is, John Travolta, he has a dominant trait for a homologous cleft chin, capital T, capital Z. Now here we have an individual, well, same individual, but here this individual may have, it's called heterozygous because they have the dominant trait. Yes, they will have the cleft chin, but they also have a trait for not having they also have a recessive trait. Here it's the same thing. And here this person does not have a cleft chin because they're homozygous for an uncleft chin. I might skip ahead a little bit where it talks about here we have a female who is heterozygous for the cleft chin, and the male who is also heterozygous for the cleft chin. And their offspring, you have one individual, one child, to say out of 4, one child will be homozygous with the cleft chin, so they will have the cleft chin. The 2 here will have heterozygous traits with the cleft chin, so they will have it, but they'll also have a recessive trait. And the 4th child will not have that cleft chin, homozygous for an uncleft chin. So that is just looking at a Punnett square. I'm sorry, I kind of moved ahead of myself. So just give me a second. All right, so go back. So that's dealing with your dominant trait or your dominant allele and your recessive allele. Recessive will always have that lowercase letter, and your dominant will have the capital letter. All right. Now the alleles that an individual possesses constitute the genotype. An observable trait is called the phenotype. So the person with the cleft chin is considered to be a phenotype because it's an observable trait. An allele is said to be expressed if it appears on the phenotype. The only way most recessive alleles can be expressed is if an individual inherits from both parents. Now, recessive traits can skip from one generation to the other. Now, a diagram called a Punnett square shows how heterozygous parents can produce a child with a homozygous recessive traits, which I just showed you. Each parent can produce go back to that chart. Each parent produces 2 types of germ cells. You have 75% will be the phenotype, which are, what did we say phenotype was? Basically, you can actually see the trait. The trait is physical. It's an observable trait. So 75% of this particular chart is a phenotype, while the remaining 27% is a recessive phenotype. You cannot see the cleft chin. Now, heterozygous individuals with normal phenotypes may be carriers of the hereditary disease or trait in forms of recessive alloys, which is that lower case, such as in the case of cystic fibrosis or even sickle cell anemia. Now, for some hereditary diseases, tests are available, and genetic counselors can have tests performed and advise potential parents of risk. So for any females or any individuals that have been around pregnant women, certain tests are taken to see whether or not the child has any diseases or traits that were passed on from parent to parent. Also, even after, if you don't have that particular test done during your pregnancy, you can have a blood test done to see whether or not the child is dominant or recessive for a particular a particular disease, such as sickle cell. If your spouse, let's get my pen out. If your If your spouse has, let's do sickle cell. Let's do S. If your spouse has a dominant trait of sickle cell, let's do ovaries. So we know this is the female. And the male counterpart is, he also has sickle cell, but has this female is a homozygous trait for sickle cell, and the male is heterozygous for the sickle cell trait. We know just by going through the Punnett square that child number 1 will have sickle cell, child number 2 will have sickle cell, and this is just bringing the capital letters into each square. This capital letter comes down, that capital letter moves over. For child number 3, I'm going to bring the S down. The capital letter is always first, just so you know. Then we have a smaller S for the 4th child. Yes. So you'll see that 100% of the children have sickle cell. 50% of them will be homozygous for the sickle cell trait. The remaining 50% will be heterozygous for the sickle cell trait. Now, let me see if I can erase this without causing any problems. What's that? Let's do this one. And this one. Alright, so let's try another example. Example 2. We'll have a heterozygous female. Let's do my lowercase s's like that. And we have an individual with no trait. He may have a recessive trait. A recessive trait of sickle cell. So what will we have? We'll bring down the S. First child will be heterozygous for the sickle cell trait. Second child will have recessive trait. So they're not going to show any signs and symptoms, but it's still there in the system laying dormant. Now, third child will also be heterozygous. The 4th child will be homozygous for the non-sickle cell dominant trait. So it'll still be there, but they won't have the symptoms. So 50% will be homozygous non-sickle cell, and the other 50% will be heterozygous for the sickle cell trait. Alright, so when we look at a gene pool, some genes have multiple alleles within the gene pool of the population. And a gene pool is a collection of genetic makeup of a population as a whole. Now we have multiple alleles, which is more than 2 allelic forms of a trait. Now, the AB blood types have two dominant alleles. We have the the 1A, the IA, and we have the IB, and one recessive little I, which is what I'm talking about right here. Let me see if it's in this one. Nope, I'm sorry. Okay, so to have type A blood, a person must have either A1, IA, or IA. So let's do that. We'll do type A blood. Oops, sorry, so do 2 dots. Type A blood. Type A blood, you will have IA or IA, that's one. Or you have IA and lowercase I. This would be type A. Now, if you were type B, I'm sorry, I'm typing all over the place. Type B blood, what would you have? You will have IB, or you will have IB and lowercase I. All right. Let's move over here. If you have type AB blood, you have IA, that's an A, and IB. Alleles. These are your alleles we're referring to. If you have a type O, what do you think type O would be? Type O, little I. So these are your alleles for each blood type. Now, the two dominant alleles for the ABO blood types are examples of alleles that are equally dominant or they're codominant. Now, the A and the B alleles, the code for enzymes that produce different surface glycolipids on the red blood cells. Now, in type AB, both types of glycolipids are present. And then in type O, neither type is present. Now, some alleles exhibit incomplete dominance, in which the phenotype is intermediate if two alleles are present. Now, we have something called polygenic or multiple gene inheritance, and it occurs when genes or two or more loci, or even on different chromosomes, they contribute to a single phenotype trait. Now, the human eye and skin color, are examples of this polygenic traits. The Pleiotropy occurs when a single gene produces multiple phenotype effects. Now, an example of this would be your alkaptonuria, and it's a disorder caused by mutation of your chromosome 3, and that blocks the breakdown of the tyrosine and results in an accumulation of a particular acid. Okay. Now, the oxidation of this particular acid, it causes connective tissue to turn gray, to blackish, and it causes degeneration of the tissues. Now, urine also turns black when exposed to air. Now, in addition to darkening, damage also occurs in the joints, heart valves, and other internal organs. Now, sickle cell is another example of this pleiotropy. Pleiotropy. Now, sex-linked traits are carried on the X and the Y, X or Y chromosome, and therefore tend to be inherited by one sex more than the other. So, men are more likely to inherit red-green color blindness or hemophilia than are females. Now, these phenotypes are based on alleles on the X chromosome. A female has 2 chromosomes, so they may be heterozygous, whereas a male has only 1 chromosome and normally expresses any allele found there. Now, the X chromosome carries about 260 genes, most of which are not related to sex. The Y chromosome has very few genes, so that most sex-linked traits are associated with the X chromosome. Now, in some cases, the genotype does not produce the expected phenotype. Penetrance refers to the percentage of population with a given genotype that actually exhibits the predicted phenotype. The penetrance of the polydactyly allele may be 80%, indicating that 80% of those having the polydactyly genotype exhibits extra digits. Environmental factors play an important role in gene expression. At the very least, all gene expression depends on nutrition. Now, dominance and recessiveness are unrelated to how common an allele is in the population. Type O is the most common ABO type, blood type in North America, and polydactyly is rare. The fast-growing field of epigenetics is a study of hereditary changes that are not caused by mutations in the DNA. DNA methylation is a common way to alter gene expression without altering the DNA sequence. Inappropriate DNA methylation has been implicated in cancers and hereditary diseases such as Prader-Willi syndrome. If epigenetic changes persist over multiple generations, your phenotype may be a result of environmental influences on your grandparents, just as environmental influences on you might affect your grandchildren. Okay. So let's look at cancers for a minute. Everybody is aware of what cancer is, and how it affects the body and the various stages. But when we look at malignant tumors, these are the ones that cause cancers. You have malignant and you have benign. Benign are the ones that are non-cancerous, and they are the ones that cause cancers. They are fast-growing, and of course they tend to give off cells that see the growth of multiple tumors. It's called metastasize. Sorry. Now we have oncology. Oncology is a medical specialty that deals with both benign and malignant tumors, but only malignant ones are called cancer. Cancers are named for the tissue of origin. It all depends which one we're dealing with. Are we dealing with the skin? Are we going to deal with lymph nodes? Are we dealing with pigment cells? Are we dealing with blood forming tissues such as the bone marrow? Are we dealing with bone or other connective tissues or muscles? Everybody, it all depends on which where the cell is originating from. Okay. Now, about 90% of the malignancies are carcinomas, and probably because the epithelial cells have high rate of mitosis, making them especially subject to mutation, and because the epithelia are more exposed than other organs. Now, only 15 to 10% of cancers are hereditary, but cancer is always a genetic disease. It is not a contradictory as it may seem. Most cases are due to mutations arising anew in the affected individual, not to genes received from a parent. Mutations can arise simply through errors in DNA replication or from exposure to carcinogens, such as radiation, dealing from UV rays or X-rays, chemicals as of just smoking cigar, viruses such as HPV, hepatitis, and type 2 herpes simplex. Now, the oncologists, which we know who oncologists are, we know what oncology is. They're especially interested in two families of cancer genes called the onconogens and the tumor suppressor genes. Okay. And onconogen is basically stuck to, it's like your foot stuck to a particular accelerator, and they cause cell division to accelerate a lot more than what it's supposed to. So if it's supposed to go a certain speed, it's going to go a lot faster than that. They basically, then we have the tumor suppressor gene. It's like they say it's like a broken pedal. So the healthy tumor suppressor gene, it inhibits the development of the cancer, which is great. A healthy TS gene inhibits cancer by opposing onconogen action, which is what you want, coding for the DNA repair enzymes and other means. Consequently, mutations that destroy the mutations that destroy their protective braking function can lead to cancer. The mutation of this TS gene, called the P53, let me see where we are. Okay, of this P53, for example, is involved in about 50% of cases of leukemia and colon, lung cancer, breast, liver, brain, and of course with esophageal tumors. Many human cancers are associated, however, not with mutation, but with aberrant DNA methylation, which can, for example, silence one's TS genes and thus turn off all protective function. Now, here's an example of what the Mills tumor looks like. It is a malignant tumor of the kidney. This is what you have occurring in children. Here's the tumor right here. Some lethal effects of cancer. Everybody knows what it can do. Replace functioning tissue in the vital organs. And they, of course, invade blood vessels, lung tissue, brain tissue, especially if it's not caught early. If it's not early, it can spread. Even if it is caught early, depending on the type that you have, it can affect the lymph node and your immune system, causing complications to other parts of the body. So if you have any questions, that is the end of our chapter 4. If you have questions, just post on the discussion board under questions and answers. All right, good luck.